The overall goal of this procedure is to study age-dependent genomic instability in yeast by combining the chronological lifespan with a set of simple DNA damage and mutation assays. This is accomplished by first sampling chronological aging cultures every 2 days and checking for viability by colony-forming unit or CFU formation. The next step of the procedure is to examine the DNA mutations by sampling aging cultures and plating cells on mutation-specific selection medium. Ultimately, results can be obtained that show changes in DNA mutations during yeast chronological aging through normalizing the mutation assay results to the viability.
This method can help answer key questions in the aging field, such as the mechanisms that link aging and genomic instability in higher eukaryotes.
To assay for yeast chronological lifespan, or CLS, first inoculate a single yeast colony into 1 milliliter of synthetic complete glucose, or SDC medium, and incubate overnight with shaking at 30 degrees Celsius. On the following day, dilute the overnight culture into 10 milliliters of fresh SDC medium to an OD600 of 0.05 to 0.1. Incubate with shaking at 30 degrees Celsius for 3 days. On day 3, when cells are in stationary phase, dilute each aliquot 10,000 times in autoclaved water. Then, remove 2 10 microliter aliquots from the flask and plate 10 microliters of each diluted culture on a YEPD plate. Incubate the plate at 30 degrees Celsius until colonies arise. The number of colonies arising, or colony-forming units, from the day 3 culture is considered 100% survival. Continue removing aliquots from the aging culture in subsequent days for plating, adjusting the dilution factors accordingly to ensure about 20 to 100 colonies in the CFU assay. Sampling is stopped when the viability of the culture has decreased below 1%, typically between days 11 and 13. Cell viability can also be studied in cells aged on plates. To begin this assay, inoculate a single colony into 1 milliliter of synthetic complete glucose medium and incubate overnight with shaking at 30 degrees Celsius. When the culture has grown to about 10 to the 8th cells per milliliter, dilute the culture with autoclaved water to 100 to 200 cells per 10 microliters and 1,000 cells per 10 microliters. For each strain, prepare a set of 8 to 20 tryptophan dropout SDC plates labeled according to plating density to ensure 10 to 100 colonies can be counted in anticipation of decreased viability during aging. Plate 2 aliquots of 10 to 30 microliters of diluted culture onto each plate. Incubate the plate set at 30 degrees Celsius for the duration of the lifespan analysis. On the day of plating, and every 2 days subsequently, remove 1 plate and dropwise add 0.5 milliliters of tryptophan to allow viable cells to grow. Incubate the plate at 30 degrees Celsius for an additional 48 to 72 hours. After 48 to 72 hours, count the CFU for the viability for the day of tryptophan addition. To assay for DNA damage and mutation frequency during chronological aging, a number of strains with special features are grown in parallel with the wild-type strain for the yeast CLS assay. Spontaneous mutation frequency can be evaluated by measuring the frequency of canavanine resistance in a chronologically aging culture. Mutations in the CAN1 gene, which encodes the plasma arginine permease, render cells resistant to the arginine analog L-canavanine. Canavanine-resistant cells will grow on arginine dropout synthetic complete medium plates supplemented with L-canavanine sulfate. Measurement of TRP-positive reversion frequency in a strain containing an amber mutation in the TRP1 coding sequence allows the estimation of the rate of base substitution during yeast chronological aging. TRP-positive reversion cells will grow on tryptophan dropout plates. The lyse-negative strain EH150 harbors a +4 shift in the open reading frame, which results in auxotrophy for lysine, and this can be reversed by small insertion or deletion mutations. The revertants will be selected on lysine dropout plates. To detect gross chromosomal rearrangements, or GCRs, a mutant strain is used in which HXT13, located 7.5 kilobases telomeric to CAN1 on chromosome 5, is disrupted by a URA3 cassette. Mutations in both CAN1 and URA3 genes render cells resistant to L-canavanine and 5-fluoroorotic acid, respectively. Since the frequency of point mutations occurring in both genes is low, analysis of the frequency of resistance to both L-cannavaline and 5-fluoroorotic acid provides an estimation of GCRs. To monitor the level of homologous and homeologous recombination during chronological aging, mutants are generated that carry either 100% homologous inverted repeats or 91% homologous IRs at the His3 locus. Recombination between the IRs allows the expression of functional His3 protein. Therefore, cells in which recombination has occurred will grow on histidine dropout plates supplemented with galactose. On day 3, when cells are in stationary phase, an appropriate amount of cells will be removed from each aging culture. Since the same procedure is used for all the assays, only one sample will be processed for the purpose of this video. Pellet the cells by spinning in a benchtop centrifuge. Remove the supernatant and resuspend the cells in 1 milliliter of autoclaved water. Pellet again. Remove the supernatant and resuspend the cell pellet in 100 microliters of water. Plate cells on the appropriate plates to select for the desired revertants. Incubate plates at 30 degrees Celsius. After 3 to 4 days, count CFUs. The mutation frequency is normalized to the number of viable cells. This graph shows representative results from a typical chronological lifespan assay. When the chronological survival of wild-type cells is compared to that of SCH9 delta, TOR1 delta, and RAS2 delta mutants in liquid culture, mutants lacking TOR1, SCH9, or RAS2 show an extended chronological lifespan. Furthermore, deficiencies in SCH9 and RAS2 show an additive effect on longevity promotion. Chronological survival in the presence of various carbon sources using the in situ viability assay is shown in this next graph. Day 1 SDC wild-type cultures were diluted and plated onto SC-trip dropout plates with no carbon source, SC-trip dropout plates supplemented with glucose, ethanol, or glycerol, or agar plates. Plates were incubated at 30 degrees Celsius. Every 2 days, 1 plate was retrieved from each set and appropriate nutrients were added to allow growth and colony formation. Age-dependent mutation frequency varies greatly depending on the strain background, genetic manipulation, culture conditions, and age. This table shows typical results obtained in a wild-type strain. Representative results of translesion synthesis is shown in this next image. The age-dependent mutation frequency increase may involve changes in erroneous DNA repair. Using nuclear extracts and various DNA templates, we can evaluate the translesion synthesis, or TLS, in the chronologically aged cells. Nuclear extracts from 3-day-old stationary phase wild-type and SCH9 delta mutant cells were analyzed. TLS products with undamaged template are indicated by solid lines, and TLS products with damaged template are indicated by dotted lines. There was no translesion synthesis observed in nuclear extracts from the long-lived SCH9 delta mutant. Free primers are indicated by the open arrow.
After watching this video, you should have a good understanding of how to perform chronological lifespan and DNA mutation analysis in yeast.
The applications. Methods described here can be further expanded to study age-dependent genomic instability. Quiescent and non-quiescent cells can be isolated from yeast stationary phase cultures using the density gradient method, combined with the mutation assays. Take into consideration age-dependent frequencies of different DNA mutations vary greatly depending on the strain background, genetic manipulation, culture conditions, as well as regrowth/gasping and extremely low survival. Pre-experiments should be carried out at time points such as the mean and maximum survival with various plating density for mutation assays to determine mutation frequency range before performing the full-scale lifespan analysis. Multiple biological replicas should be included in the study, and both liquid and in situ viability/mutation assays should be carried out to corroborate the results. Liquid aging cultures sometimes exhibit regrowth/gasping phenotype 13, which complicates the CLS analysis. Thank you for watching. Subscribe if it is useful.